All right, so I'll kick off this double feature today by introducing Eric Liu. So Eric Liu is our 2023 Vaughn Jordan intern in the plant ecology program. Eric is from Pennsylvania and graduated from Cornell University in 2022 with a BS in plant sciences. His numerous research experiences prior to coming to Archbold include working in a wood anatomy lab, a coal lab, and a herbarium. Eric arrived at Archbold in April and has been a crucial member of our team. He has a wonderful curiosity about the natural world, and especially having been here through the particularly hot summer, is superbly hardy as well as meticulous in the field. In addition, he is a talented musician, Pokemon expert, and I'm told he has a very impressive standing long jump. For his intern project, Eric was drawn to the seasonal ponds and specifically the state-listed Hypericum edisonianum. As you'll hear in his talk, this species is intriguing in part because despite its severely restricted geographic range, it is highly abundant in our area and found in a variety of habitats. In choosing a wetland species, Eric had the daunting task of navigating around the exceptional wetness at the station this season, which I'm happy to say did not derail his project. And with that, I'll hand it over to Eric.
Right. Hello. My name is Eric, as Aaron so graciously introduced me. I'm the Von Jordan Plant Ecology Program intern, and here's my project, the Microhabitat of Hypericum edisonianum. The slide will turn. I don't think it is working. Oh, sorry, enter. Enter. Apologies on that. Okay. Here. Apologies for that. Yeah, so my first days on station, as Aaron described, I saw this woody, shrubby plant that existed in the seasonal ponds here on station, and I was intrigued because not only were they in the ponds, but they were also along the roadsides, which was not usual for, especially for something that is state listed to be expected. You'd expect endangered plants to have a very restricted area where they can be. This is a state listed plant, though it is also a candidate for federal listing. It's a Lake Wales Ridge endemic, almost. It does slightly go off the ridge for getting to areas such as Buck Island Ranch, which is off the ridge. But yeah, it's a small woody plant with these beautiful four-petaled yellow flowers. And there's not a ton of information about this species. The most of it came from Warren Abramson with data collected in the 1990s out of actually here, Archbold Biological Station. And in his publication, he basically described this layout for a pond, where we would expect cutthroat grass in the center, represented by green. It's also interesting to note that cutthroat grass is another endangered species that we have here at the station. And then outside of the center of that cutthroat grass, there was a ring of Hypericum. And outside that ring of Hypericum, there was another ring of palmetto about the pond margins. Ideally, there's water throughout these in the wet season, but in the dry season, this can get pretty dry to sometimes completely dry. This is the established pond layout that was published. And here is a picture of something that looks kind of similar to that, where we have, if taken from the center of the pond looking outwards, we have the center of graminoids, in this case, graminoids being grass and grass-like plants. Because in this picture, there actually was not as much cutthroat grass. It was xyrus instead, which is a different graminoid-ish plant. But you can see as we move towards the outside of the pond, we've got this ring of Hypericum in the foreground background area that the xyrus bleeds into. And then by the third arrow, we've got this ring of palmetto at the pond margins. Also, that's not the only place that I found Hypericum. This picture is at a pond edge, and this red circle here shows another spot, another Hypericum plant. There are granules present. In this case, there is the cutlery grass, but also there's palmettos, as we expect at a pond margin, and there are also gallberries, which was not really mentioned by Warren Abramson. So this is another slightly different area where we found this Hypericum edisonianum. But also, there's more. This picture was taken on the reserve, which is a highly modified spot owned by Archbold. Once again, the Hypericum is circled in red, and this area has a lot of bare sand, and it's with palmettos. This area also is nowhere near any ponds, not at all. Historically, there may have been ponds here, but at the present moment, and for a long time now, there had not been ponds here. There's also this random herbaceous plant, which was not expected to be found near Hypericum. And this picture, is also taken from the reserve. You can see it's a wide open, grassy, plain almost, not really plain, but like flat area. And these brown dead stems are actually dead Hypericum plants. It's possible that they died from a prescribed burn or maybe disease. It's hard to say, but they do have little plants coming up from underneath them, so they're not done yet. But what was most interesting about this is that the brown stems do not extend across the entire landscape. If this entire region was good for the Hypericum for whatever reason, we would expect it to kind of be covering this entire place, kind of like what the grass is doing. So there must be something that is preventing Hypericum edisoniana from completely taking over this area. From a visual glance, it's not really easy to see. And so I'm kind of looking at, here's some questions. For a plant that has such a restricted geographic range, why is it able to persist in such a variety of habitats? I showed 4 different habitat types that Hypericum was in, and it seemed to be doing just fine, or at least was existing in all 4 of those. To break down this question a little bit further, I'll split it into 3 sub-questions. 1, what are the microhabitat factors which affect the presence or absence of Hypericum edisonianum? Is there evidence that it is a strong or weak competitor? The idea behind this is that maybe the graminoids do best in these wet areas of the ponds, and the palmettos can do well in the dry areas. And maybe at each of their respective margins, this is where the Hypericum can grow, where neither the grass, the graminoid, or the palmetto does as well. And it takes that niche there, being a poor competitor. And then 2 and 3, how does habitat modification affect these factors, and how does habitat modification affect plant performance? I had mentioned that the reserve was a highly modified area, and on station 2, a lot of the ponds that we have here are modified in one way or another. And anthropogenic influence is always the question that's kind of in mind for us, or whether plants are going to be able to keep up with what we're doing to the planet or not. The difference between 2 and 3 is that 2 kind of talks more about where Hypericum can exist, whereas 3 is asking how well the Hypericum does when it does exist. To answer these questions, I took the approach of an observational study, and I collected 15 ponds across the full biological station. Of these 15 ponds, I picked 3 each of 5 different modification categories. These modification categories being ditches, roads, road ditches, which are essentially deeper roads, trails, and unmodified ponds. The ditches, roads, and road ditch ones I classified as high modification because there's more modification that went into making these ponds what they are, and low modification, which are the trails, which are essentially just foot trails people or perhaps animals have used to traverse through or near the ponds, and unmodified, which are, as the name suggests, unmodified ponds. So at each of these ponds, I sampled 7 quadrat, 1 by 1 by 1 quadrats, 1 meter by 1 meter quadrats. To get these quadrats, I first took a transect line from relative to the pond, the outermost Hypericum that I could find, and the centermost Hypericum that I could find. And with that transect line, I then went 1 meter out from the innermost Hypericum and 1 meter in from the outermost Hypericum and put the quadrat there. And then somewhere in the middle of that transect line, I also put third quad for, and these in red are my presence quads. Now, in most of the ponds, there was not a full ring of Hypericum around the center. There were areas such as in this blue area down here where there was actually just not any Hypericum for whatever reason. So I also mimicked my presence quads with absence quads, basically translating the transect line down into the absence areas. So I'll then have an innermost, an outermost, and a centermost absence quad at roughly similar distances from the center of the pond compared to the presence quads. And then thirdly, I had 1 demography quad per pond, and this is basically where the Hypericum appeared to be performing very well. I took into account things such as percent cover, the number of reproductive structures, how big the plants were, and how healthy they looked and less diseased. And through all these, having relatively consistent things that determine this demographic quad, I was able to compare between ponds. Now, what was I looking for in each of these quads? In the presence and absence quads, I was looking at biotic factors, namely percent cover. And I had basically 4 target species. I had the Hypericum edisonianum, of course, because that's kind of the plan of the study. I had the graminoids, whether they were cutthroat grass or xyrus, or very occasionally there were other things that were in there, but I would count these graminoids. I also looked at gallberry, personally observed to be in some of these areas, and look at the palmettos, because we expected the palmettos to be, especially on those outermost areas. I also looked at canopy cover, which I measured via densitometer. I also looked at abiotic factors, such as sand, percent cover of sand versus litter, and water. I also looked at the pond modification history, as I previously mentioned. In the nomography quads, I looked at the biotic factors, once again, percent cover of Hypericum in the quadrats, the number of stems, which I broke into adult and juvenile stems, and the number of reproductive structures. So this would be basically buds, flowers, and fruits. I also looked at the pond modification history, and I took the GPS location of each of these demography quads. So this would be getting the easting, the northing, and the elevation. So after going through all these and with a month or so of data collection, what did I find? So here I have 2 plots. On the left one, I have the percent cover of graminoids on the x-axis and percent cover of Hypericum along the y-axis. And we can see that there's a trend, that a slight negative trend, where with increasing graminoid cover, we have decreasing Hypericum edisonianum cover. Statistically not significant, but it's still an interesting trend nonetheless. And so then I looked at, well, maybe the grasses are doing better where there is no Hypericum. There's maybe some competition between these 2. And this brings to the second plot, where I have the quad type on the bottom with the presence quads, which is the ones that have Hypericum in them, on the left, and the absence quads, which are those white boxes on the previous slides, where there was no Hypericum, and then percent cover of graminoids on the y-axis. And here, there appears to be a higher amount of graminoids in the absence quads. So this suggests that where there is no Hypericum, cutthroat grass does do better. Findings are not statistically significant, but again, this is an interesting trend, or where the means are over double. Yeah. Secondly, I have this set of plots showing that modification appears to reduce the percent cover of Hypericum edisonianum. On the left, I have the modification type along the x-axis, and I have percent cover of Hypericum edisonianum along the y-axis. And you can see that in our ditches, the road ditch, all the percent cover mean of Hypericum seems to be less 10 or less, whereas the trail on unmodified ones, it's definitely higher. And especially in the unmodified ones, the variation between high and low point is quite different. However, once again, the statistics were non-significant. But when I put them into their modification category, remember that this is the ditch, the road, and the road ditch together as high modification, and the trail and unmodified together as low modification. We actually do get statistical significance with the low modification ponds having about 2.5 times the amount percent cover of Hypericum edisonianum compared to the high modification ones. Thirdly, shows that modification categories have variable effects on the reproduction. These were the demography plots that I looked at. So I went straight to the modification category for these. And high modification refers to the fact that there's going to be low amount of juveniles in each quad compared to the low modification ones. So again, the variability is a lot higher. And then for reproductive structures is actually the opposite trend, where the high modification ones has this high variability and slightly higher mean compared to low modification ones, which has much lower variability in reproductive structures. So in summary, Hypericum edisonianum and cutthroat grass may have a microhabitat overlap, which may be contributing to competitive exclusion. Other species that I looked at, which were the gallberry and the palmetto, did not actually appear in my quadrats as much as I expected them to. So the data from that was sparse and not very usable. Secondly, pond modification prefers to reduce the percent cover of edisonianum, which makes sense. In more disturbed areas, spare plant might not be able to establish itself as much or survive as much, or maybe gets dug up directly. Who knows? And thirdly, there may be potential trends on pond modification or reproduction. We definitely saw that there was more variability, and there's a little bit of a mean difference, but what the trends exactly are is not exactly clear. So taking it a little next step, level of modification may matter more than the specific modification type. This went to the idea of the fact that with the modification categories, all 5 of them listed out, those findings were statistically not significant, but when I put them into the categories, they did become statistically significant. So maybe it doesn't matter if the area was dug to become a road or dug to become a ditch or was a deeper road than, say, but those kind of all have a very similar effect as each other. But there also is definitely more work needed to determine competitive relationships and capabilities of Hypericum edisonianum, especially with cutthroat grass, as shown by the first couple of my graphs. Cutthroat grass does seem to do better where Hypericum edisonianum is not. Similarly, when there's cutthroat grass, Hypericum edisonianum seems to do worse. But we don't know exactly for sure because of this low sample size, but there's definitely a trend to be explored further. And as I said, additional sampling could reveal statistical trends. I only measured 3 of each type of pond. So with more sampling, maybe that gives us more power in our statistics, though it also may be difficult because, for example, I believe there were only 4 or 5 ditches on station. There's only so many more ponds that are actually available to be sampled. So there might be some mysteries that are going to be very hard to uncover. So in a broader sense, what does this mean? The observed Hypericum edisonianum habitat does indeed to be broader than previous studies have shown, including mine. My study was done exclusively on Archibald Biological Station. Some of my preliminary observations were on the reserve, for example, and I didn't even visit the ranch. So there's possibly even more data, definitely is more data to collect that this could add to and give us a more holistic understanding of this amazing plant. And the idea is maybe these areas that are outside the ponds are just marginal habitats for Hypericum. Maybe the reason it's restricted is, or rather the reason that we're seeing it in all these things is this plant is trying to branch out in areas, but it can't really quite get past just these marginal areas. Maybe that's what's happening. And again, there's still a lot of work to be done between Hypericum and cutthroat grass. Maybe these both endangered or nearly endangered plants are competing with each other, and it'll be cool to see how they are. Because usually, you would expect like something an invasive or a very dominant plant to be kind of beating up on the smaller guy, the more endangered one. But if these are both endangered, it's kind of interesting to see what we'll find in the future. So yeah, thank you to coming for my talk. I'd like to thank everybody on the slide. There's definitely more people not on the slide that have contributed to my internship as a whole, my study, my project, just living here. Like it would not be the same without each and every one of you guys. So thank you.
All right. Do questions? Hopefully we can hear on this one. This round.
Do you have any idea why it might have been that you observed more reproductive structures in the highly modified habitats?
Yeah, so there's some ideas, like perhaps the plants there, because the number of juveniles was so low, so maybe the plants that were there are adults, so maybe they're just the best or the strongest of them, so they can produce more. Or maybe they're under stress. Some plants can flower more when they're under stress. They're kind of anticipating that they're going to be gone before too long, so they're trying to get set seed as much as they can before they go. So either one of those could be an answer, or maybe there's another thing so those are my ideas.
Hey,
Yes.
Eric, great talk. So my question is, when you were measuring the ponds, you mentioned the competitive exclusion, but you didn't mention, is there like a difference in depths or anything of the water, like where you're seeing more of the cutthroat versus, I'm sorry, I'm not going to try to pronounce that scientific name.
The
And
Hypericum?
whether it's you think it's competitive exclusion, or maybe there's even differences within the microhabitat that they're just better able to grow in.
Yeah, so there could be some of that, but also the ponds were not really consistent with each other. There were a lot of little details that were like one of in a pond, or like this pond had this specifically, but not necessarily all the other ones. Like there were a couple ponds where the Hypericum actually ended up in the center of the pond, which was weird. And the cutthroat grass, in many of them, was not restricted to only the center, but it was kind of in like half of the pond. But it wasn't really like a innermost or outermost thing. So there definitely, I would anticipate there probably is some sort of microhabitat differentiation, but there wasn't a lot of consistency. So it was hard to make or show a trend. If that answers your question.
I'm wondering how fire affects the species, and in your study, did you control time since fire, or did you note that in any way?
Fire might have an effect. I didn't really look at fire too much, though I do remember that there was one pond that had a road that went through it that basically split two units. And I was told after I went and looked at it, that one side had been burned much more recently than the other side. And there was actually a pretty stark difference where one side had a lot of Hypericum and Xyrus, where the other side had a lot of grass, actually. It was very interesting to see that difference. But again, that was another one of pond. So it was very hard. It's hard to draw conclusions from that.
All right. We have a few questions online.
Yes.
This was from Eric Mangus. There's been a few specific studies, like Gail Vander Kerkhoff did a PhD showing various effects of disease, and some other ones by Stephanie Kuntz. How do your results compare to these studies?
I did not see a ton of evidence of disease. There were a couple galls on scattered plants, but I didn't see a whole lot of, let's say, like swaths of dead Hypericum, like fourth picture of on the reserve. So yeah, I didn't look at disease too much, really at all. But it would be interesting to see if disease maybe helps it or hurts it in cutthroat grass areas, or maybe it's confounding factor or something.
Another question, did you find any new patches on station?
New patches on station. New patches as in where areas where we haven't seen Hypericum before, I'm assuming. And actually, I went to each pond with the assumption that there would be Hypericum there. And I used that as my baseline for finding Hypericum. To my knowledge, there wasn't a set database of exactly the locations of all the stands on station. And so, yeah, actually not every pond had Hypericum in it, which was interesting. But yeah, I guess I may have found new ones.
I'd agree. It's much more interesting when you didn't find it at the pond than where you did of at every pond. And one more online question. This is from Alan Liu. How can we use the results of this study to help preserve both Hypericum edisonianum and the cutthroat grass, given that they're both under threat?
Yeah, so I've shown that there are some factors, such as mostly the modification, that appear to have a negative trend. So, we have our bread and butter. Let's develop less in areas where endangered plants live. That's not necessarily new, but this does show some direct evidence that perhaps this is modification, and driving trucks through ponds might have a detrimental effect on these. And maybe there's a confounding factor that's affecting both the Hypericum and the cutthroat grass that we're not even necessarily aware of. But yeah, it opens the door to be like, ooh, there's more things that we can look at, I would say.
We'll do one last question.
I was wondering if you observed much hog rooting when you were going at these ponds, and if you would consider that to be a modification that could impact these species.
Yeah, I actually did not find very much hog rooting. There was one pond, it was a ditch, and it was on the northeastern side of station that did have hog rooting in it. That was very obvious. But for the most part, I actually didn't find a lot of hog rooting, which was interesting because before I went out and talking with Betsy, she also expected there to be a lot of hog rooting in a lot of these ponds. But I didn't really find that, which was interesting.
All right.
Lucky or something.